Dear Jacques, ladies and gentlemen, I'm pleased to be with you today to celebrate that day, and I continue to look back and then towards the end also to look forth. Jacques already reminded me that he came to the Laboratory of Biophysics in Geneva, where I was working on my PhD studies. I had before studied here at the ETH natural sciences. I regret that that curriculum doesn't exist any longer because it gives a wide introduction to mathematics. It gave to mathematics, physics, chemistry, all of life sciences, earth sciences, mineralogy, and so on. I always encounter important contacts nowadays, for example, in structural biology of proteins, crystallography is important to have some notions. So is it in physics. Actually, in the ETH, I decided to make my diploma work with Paul Scherrer in nuclear physics, experimental. And towards the end of this, when I had accomplished my examination for the diploma, I went to the office of Paul Scherrer because I knew that he had usually offers for jobs. I wanted to get some job. And when I came, he said, well, you are from the section of natural sciences, section 10. I have something quite good for you. At the University of Geneva, they have an old kind of experimental electron microscope. At that moment, electron microscopes were not freely available on the market. Trüb-Teuber, a Swiss firm, had actually constructed some and needed the help of physicists. And some institutes of physics in Switzerland had accepted that, and then they received for free these experimental microscopes. And I came to Geneva to take care of one of those and started to look at bacteria, at bacterial viruses and so on, because they had there already good contacts with the so-called phage group, which was actually around people like Max Delbrück, Salva Luria, and so on. And Jean Wegley was the contact person, a physicist who had actually given up the direction of the Physics Institute when he was 50 years old and went to Caltech to work with Max Delbrück. And Jean Wegley, being a citizen of Geneva, came every summer to Geneva, worked in the lab from 5 in the morning till the early afternoon. Then he disappeared. But when I came in much later than he already knew what the results of my experiments of the day before were, and we had very good contacts. So that's how I came into getting some first results on microbial genetics. I will cite one of those. And then usually at that moment, the people working in microbiology usually reported their results at the annual meeting of the Society of Microbiology of Switzerland. So these are other ways in which I run into Jacques Nürsch. We had good contacts, and we mutually followed the advance which I was working in prokaryotic microorganisms, while Jacques was working rather in eukaryotic microorganisms. But there were good contacts there. And since ever, we had become good colleagues. And when we then finally both moved to Geneva to Basel in the early 1970s, Jacques at the pharma industry, me at the Biozentrum, which was started in 1971. We mutually profited from each other. Thanks to these good contacts with the scientists at the pharma industry, we profited a lot in our teaching, and we could place some of our students, also PhD students, diploma students, to their labs, and were sure that we had good contacts between students and professors. So this is really, thank you very much, Chuck. You were very efficient there to organize that, and some of the younger people moved into that also. So we, from the beginning at the Biozentrum, profited a lot to have these good contacts. So let me go then to this is kind of history of science, what you see here. We can remind ourselves that in the mid-19th century, there were actually three important new developments starting. Darwin with the evolutionary biology, Mendel with genetics, and Miescher with nucleic acid biochemistry. The advance in these fields was very slow. Genetics was mainly done with higher organisms, animals and plants, and that lasted till around the mid-20th century. When then, in fact, it became clear that Mendel working with variants and Darwin working with variants, that these were actually genetic variants. So this gave rise to the modern evolutionary synthesis. About at the same time, microbial genetics started. I will go into that in a little while. And in the meantime, also Watson and Crick, after a long period of not very intensive research, came up with the double helical structure of the nucleic acid DNA, which, as we will see in a while, microbial genetics showed that it carried the genetic information. That gave rise to molecular genetics, and it is now time, and I will talk on that, to make another synthesis, molecular synthesis, to lead to molecular evolution. Let me see. Now, to the mid-1940s, it was becoming clear that not only higher organisms, like animals and plants, had genes, but genes were also present in single-cellular bacteria and in viruses, in their viruses. So then it was possible to starting to do genetics at that simple level, reminding you that bacteria have the advantage. They are single-cellular, haploid. So when there is a mutation, the expression is relatively fast. And what is very nice, E. coli bacteria, with which much of the genetics has been done, under laboratory conditions at 37 degrees, duplicates in 30 minutes. So in one day you have tremendous populations, and that facilitates enormously research, population research and population genetics. It was then seen that work done in the lab of Avery in New York, that with pneumococcal bacteria, you could extract, make different extracts, DNA, RNA, proteins, and so on, and mix this with other bacteria which had other traits, other genetic traits. And then some of the traits from the donor bacterium, here drawn in blue, penetrated and transformed these bacteria. That works only with DNA. It doesn't work with RNA from the donor, nor with protein fractions and so on. And it is clear that was then very clean experiment to show that it is DNA that carries genetic information. You can imagine that was a surprise, and not very many biologists understood what that means. Fortunately, just a few years later, the double helix of DNA came up, and it was clear that the genetic information is a linear arrangement of nucleotides. I always try to compare that linear arrangement of nucleotides in the genetic script with our script. We have more letters than 4. DNA only has 4 letters, but still, that is the way how information is stored in nature and in our cultural books and so on. Joshua Liderberg and some other collaborators about at the same time could show that E. coli bacteria, some E. coli bacteria which they had in their hands, actually were able to transfer genetic information from a donor cell into a recipient cell by intense cell-cell contact. This was then called conjugation. And the interesting thing, you see there is an arrow here, and the arrow actually is the driver of that transfer. Fertility genes are there. And depending on where that fertility factors are integrated in the genome, of course, then there is a linear transfer of genetic information drawn by this arrow, and you can at any time, by shearing forces, break the bridges and show how many genes were transferred and may show up in the progeny of that green cell. So that was an indication that its genetic information is stored in a linear fashion, which then was confirmed by the knowledge on the double helical structure of DNA, which are long filamentous molecules. And at that moment, starting from the 1950s, I think then it was accepted that indeed DNA carried genetic information. Norton Cinder, a young scientist working in Joshua Liderberg's laboratory, had the task to see whether Salmonella also makes conjugation. And it first seemed that the answer is yes. But when then they looked at more carefully, they had seen that often the bacteria lysed and produced viruses. These Salmonella bacteria were carrying actually proviruses and could be spontaneously induced and produced viruses. And at that moment, some of these viruses with low frequencies incorporated blue DNA and gave that upon infection to another cell which had other genetic traits, and then the blue trait started to express its functions. So this was then called virus-mediated transduction. And here you see that a virus is a gene vector, while here the fertility plasmid is a gene vector. Now, this is one of the experiments which I carried out at the University of Geneva in the late 50s, and which gave rise to my PhD studies. In E. coli bacteria, which also was used by the Lederbergs, in fact, there is also a provirus called lambda. And that provirus then could be shown that it sometimes integrates by site-directed, site-specific recombination into the genome, most of the time at the same site, and spontaneously or by, that was a marvelous thing, by ultraviolet irradiation, the virus was induced to come up again and excise and start to multiply and give viral progeny. So that was a very nice experimental system. But Morse, working in the lab of Joshua Lederberg, had shown that lambda viruses sometimes transduce gall markers. Now, gall markers, by chance, were nearby that site on which the virus usually integrates. And actually, I was studying to see whether these viruses, which carried gall fermentation markers from the host cell, these could replicate only in the presence of an active, non-recombined hybrid virus. But that could be done, and it was clear that this kind of virus is defective, and I had the task to see whether any visible structure of viral structure came out from these cells when the viruses were produced, and I couldn't see anything in the electron microscope. And at that moment, I shifted over to do genetics and never went back to the electron microscopy. So we both finally, at that time, were really bona fide microbiologists. I could show that, in fact, this hybrid here had lost behind, you see it here, some of the viral functions for head production and packaging, and instead had a segment with the galactose fermentation markers from the host cell. I should say here that what you see here is one way of lysogeny and transduction. This is called specialized transduction. And in fact, we could also show already at that moment that each time such kind of illegitimate excision is done, it's done at a different site. So there are a lot of possibilities. And sometimes you leave more viral gene behind and integrate less. Now, of course, if the recombination creates a genome which would be too big, you cannot package it in the viral particle. So you wouldn't see those. But by density gradient centrifugation, novel lambda transducing viruses, each one has another buoyant density. I must say, you see here, I call buoyant density. That was just a few years after this methodology had been introduced. And I attach big importance that novel research methodology helps to get step forward and get new insights into the secrets of nature. You remind Sinder's lab with salmonella, experiments with salmonella, which also gave transduction. This works by a completely different way. In fact, these phages, which was called P22, and later on E. coli phage P1, they do not integrate in the genome usually at a particular site, but they are maintained as a plasmid and replicate, and when you induce, the plasmid starts to replicate. Towards the end, when it comes to packaging DNA, they look to start the packaging at a particular site, nucleosequence site on the DNA. And some of these sites, these are short sites, happen to be also on the bacterial genome here. So these phages, P22, P1, start not only to package viral DNA, but in some of the particles, they package a segment from the DNA, just a headful, so as much as goes into a head. And this is the generalized transduction. I will tell you later on, nature always, for the same purpose, to transfer horizontally genes from one cell to another. There are various ways to do so. And if you study only one system, you may be seeing how it's done there, but in other systems it's done quite differently, and still the result is quite similar. So this is, for me, was a general insight which could be largely confirmed later on. Factors limiting gene acquisition. This horizontal transfer by transduction, conjugation, and transformation, these are all rare events. Nature cannot afford to bring in all the time new information. Nature cares for having a certain genetic stability in the population. So surface compatibilities, for example, in the conjugation, cell-cell contacts must be compatible. Then when DNA enters, restriction modification systems start. I will come to that just in a moment. Functional compatibility, the DNA which was transferred into a recipient cell must be propagated either as a plasmid or integrated into the genome. And when it becomes expressed, then the harmony of the cell must not be destroyed. Otherwise, the cell has no good survival. So nature uses a strategy of acquisition of genetic information in small steps. That's what we can learn from these considerations. A little bit more detail on restriction modification systems. We run into those in the early 1960s when I came back from my US postdoc again to Geneva, being paid by a big national credit for the development of atomic power plants. Most of that money, of course, was meant for the physicists, and Edward Kellenberger had proposed that, in fact, some of that money, small amount, should be spent for biosafety studies on the effect of different kind of radiations on living beings. So we did it, of course, on bacteria and viruses. And I ran into a phenomenon which has just been described a few years before, which was called host-controlled modification. And the phenotype is such that, let's say K is a strain of E. coli bacteria. If you take the virus, you have 100 or 200 particles progeny, you can reinfect another cell, produces 100 or 200, you go like that. It's always the efficiency of infection per particle is 1. I have another strain here I call that 0, which can also still be infected without any restriction. The progeny, however, although it can go back without any restriction on the same cell, but when you go back to K, the efficiency is only about 10 to the minus 4. This is when you measure that with a multi-cycle lysate. Usually growing viruses, you grow it for a few hours in enough cells, and then you go like that. Then you have 10 to the minus 4. At that moment, I made a critical experiment, taking just one cycle of that lambda k in 0 and went back. It was not 10 to the minus 4, it was 10 to the minus 2. That was the time, 1960, when just Messels and Stahl had studied semi-conservative DNA replication. And I wondered whether that about 1% of viruses had still perhaps only one strand of parental DNA from K. And I could, by various ways, I could show that this was indeed true. So when DNA replicates, of course, from K, then here in that cell, one, at most two progeny of an infecting virus may have still one strand parental and one newly replicated. And those are actually accepted. So I will show you how the explanation is. We had been working with K, and we then started to investigate how that functions, and some years later it was possible to find the answer. There are, for all of these restriction modification systems, which has been studied so far, there are particular sequences. AAC, then N6 means any nucleotide, doesn't matter which ones, but then you are again on the same side of the double helix, GTGC. And you see that I put some stars here. The stars actually, could be shown that modification is attaching a methyl group at one of the adenines here and this adenine here in the two strands. And if this methyl group is not there, the enzyme still binds to the site, stays there, and ropes through the DNA from the drawing is a little bit not absolutely correct. From both sides, the DNA, so it's a migration from that site. And the enzyme still sticks there, but you see in the electron microscope these loops of DNA growing. And since on longer DNA molecules, there are, of course, that site can be down here again, and at some other time, the enzyme gets activated, starts to rope through the DNA, and it progresses to both sides. And when these loops run into each other, it gives a stop, and the DNA breaks there. So that's quite intelligent from nature, because if you transfer foreign DNA into a cell, you never cut at the same time at the same place. There is always some guarantee that you don't cut an interesting gene into fragments. While in the type 2, which were originally found with Haemophilus influenza by Ham Smith, it's again, you look at a particular site. Here it's GAATTC, symmetrical, with the methyl group A here. And if the methyl group is absent, the cutting is right in that sequence with staggered ends. And those are the enzymes which you buy nowadays and do the experiments. Of course, you could also do experiments with that, but it's not so pleasant to have cutting all over. And these fragments can be separated by gel electrophoresis according to more or less to their lengths, sorting it out and so on. Now I could mention here that when it became clear that viruses are gene vectors, people started to wonder whether it would be possible to integrate in the laboratory, in vitro, segments of DNA, because it was also clear at that moment in the 1950s that you couldn't study these tremendous long genomes at once. You should take gene by gene, and it was hoped that one could then analyze sequences, analyze functions. At that moment, the people were forced to take the DNA, to cut these long filaments of DNA by shearing forces. And they tried to incorporate that into natural gene vectors like viral genomes. But it was hard and didn't give much results. But when then the restriction enzymes type 2 became available, all of a sudden that was completely different. One could, starting to make projects to sort out particular segments from a genome, integrate that into cut DNA, and I have symbolically shown that here. So you take the donor DNA, you cut it with the restriction enzyme ART1, for example. You sort out these fragments, take one of the fragments. If you have a vector which also has one cleavage site for EcoR1, you just try to incorporate that here. If that doesn't cut an important function of that virus, then you can transform that into a cell, and if it grows either as a plasmid or otherwise, then you have within one day a large population. You can extract again the plasmid, cleave it with the ACOR1 and sort out the red fragment and analyze it. And that's how originally sequence analysis and functional studies were then becoming very intense in the early 1970s. I had organized already in Geneva in 1972 at the meeting center of the Leuenberg, belonging to the Protestant Church. I had organized a scientific meeting on restriction modification systems, on which also American colleagues participated. And when they came, they said, well, good, you have an interesting program, but we should also take some time to discuss conjectural risks of that methodology, putting DNA from different sources together. And we did so one full evening. We didn't come to terribly important conclusions, and it remained within our group. But some of these people, who as citizens, in 2 years later, published a letter to Science that this is really something which the scientists should really take care of very carefully. And it came to the Asilomar conference in February 1975, on which several Swiss scientists also attended. And one of the conclusions was that there was obviously short-term risks, which were due to toxicity, pathogenicity, allergenic effects, other harmful and undesirable effects, depending on a particular gene function, which it was clear that could be studied under conditions which were available from medical microbiology, because if you give a sample from, you are sick and a sample is being studied in medical microbiology, the people doing that work should take care not to infect themselves. So it was clear that that can be done, but it was said that if at some later time there are plans to liberate that by purpose into the environment or by accident. Then we don't know how that new hybrid would behave with regard to transfer to other living beings. Biological evolution was here questioned. Me and other colleagues at that moment really realized that one should bring Darwinism to the molecular level. And we started to do experiments and collected knowledge from experiments which had already been done previously. I just remind you that the neo-Darwinism, the Darwinian scheme for biological evolution. Biological evolution actually would not be possible without having occasional genetic variation, or you can call it mutation. Different alterations in the nucleotide sequence of the DNA. That drives evolution, while the direction of evolution is done by Darwinian natural selection. And just reminding you that living beings in nature are building ecosystems. No living being is alone there. I carry with me more microbial cells than human cells. It's a tremendous thing. We are ecosystems, wandering ecosystems, and so do plants and everything. So we live there. And when you do with clean cultures in the laboratory, of course, it's not really mimicking nature completely. There are other things going on. But nevertheless, I think it is important, one must be aware that the biological environment plays a role in natural selection and the physical-chemical environment. So then what we can ask now, how does nature proceed to get genetic variations? But not too often, because you have to be careful not to destroy the genomes. The conclusion, and it's not the time today to give you experiments. My intention is to give an overview. And I collect here knowledge which was then becoming available in the course of a few decades, that there are multiple sources, very different sources for spontaneous mutation without doing actively mutagenesis, applying mutagenesis in the laboratory. Some of these are replication infidelities. Some others are driven by recombination enzymes. There are various recombination enzymes already in bacteria, well studied. Then horizontal gene transfer, conjugation, for example, and viral-mediated transduction. And there are, of course, natural mutagens, internal in the living being, and external ones, for example, X-ray irradiation. And if you reflect, you have, in fact, nature uses 3 different strategies for genetic variation. One is local. One or a few letters are being changed. DNA rearrangements, segments of DNA are organized within the genome differently. And I like to show just a few examples. Local sequence changes. I'm sorry. Yeah, okay. They are a bit too close here. Local sequence changes, nucleotide substitutions, small deletion, small insertion, just a few nucleotides, and so on. And that can be an improvement by chance of available biological function. This is not reflected by nature. It's kind of trial and error process. And only very occasionally can be a source of novel activities. Actually, the chance for finding those is very small because it only starts to function if natural selection works on it. The molecular clock, which evolutionary biologists use to see when in the past two different types of organisms diverged from each other, still works on that. It wouldn't work on the other two strategies. The DNA rearrangement is recombination between segments of homology by homologous recombination, transposition of mobile genetic elements within the genome, site-specific reshuffling, and I must say that in most of the site-specific recombinations, there are also so-called secondary sites rarely being used, but which can give rise to novel rearrangements. And that can be an improvement of available capacities. For example, by novel combinations, if you have a segment from one gene fused to a segment of another gene, rarely, but occasionally can give a new function. Operon fusion would be providing a reading frame with another control element for gene expression, and so on. And finally, acquisition, that's clear. You acquire something which you hadn't before. It's a sharing of successful development made by other organisms. And I think that these two natural strategies can contribute to explain at least some of the emergence of novel properties, which evolutionary biologists always wonder about how this came about. I make some conclusions here. Recombination systems, restriction modification systems, and repair enzymes and so on are in fact in microorganisms not essential for life from one generation to the next, from one cell to produce two cells. But they are evolutionary elements. So I call these evolution genes. They can be either variation generators, many recombination enzymes do that, or modulators of the frequency of genetic variation. For example, restriction modification keeps horizontal transfer at very low level. Then we do know that non-genetic elements, of course, obvious environmental mutagens, random encounter of a natural gene vector virus with a particular cell can give a mutation, and most importantly, chemical instability and structural flexibility of biological active molecules. Time is too short to explain you that, but I came to that idea in studying what Watson Crick had already discussed in the 1953s on mispairing due to tautomeric forms of nucleotides. And I think that some biological active molecules also can. One usually believes that proteins are active in their most stable form, and people are glad to have three-dimensional structures and trying to see what the functions is. But for rare functions in nature, you may depend on metastable forms, which are short-living. One should keep that in mind from the point of view of structural biology and biochemistry. So that brings the Darwinian theory at the level of molecular processes, and most importantly, we learn that natural reality takes actively care of biological evolution. So this is a new insight which has cultural values for us. I will come to that in a short moment. So our genome, the microbial genome, our genome, plant genomes, have a duality. Many genes are for the benefit of the individual lives. Housekeeping genes, accessory genes for use under particular life conditions. For eukaryotic organisms, developmental genes. While evolutionary genes work as generators, as I explained, for occasional nucleotide sequence alterations and gene acquisition. This is the presence of these evolution genes, we assume is by second-order selection. That's a long-term, and if a population acquires these capacities, it can evolve better and is, for this reason, among us today. Otherwise, it may have disappeared already since a long time. And this is a source of biodiversity. And it's also a guarantee that nature can undergo evolution, a guarantee that in long terms, not in the course of our lifetime, but in millions of years, biodiversity is reconstituted all the time. So that's a good message for long-term development. We come back to genetic engineering, which I mentioned before briefly. Now, what one does today, one can, of course, take here a particular cell under study. We are aware that bacteria have a genome corresponding to about E. coli, about the Bible. Yeast has already 3 times more. Higher organisms, several hundred often to 1,000 times. Human genomes corresponds to about 700 Bibles, just one genome in each of our cells. You can take out selectively a small part, mutate, reintroduce it, and usually that functions only if the alteration is very minor. You can horizontally transfer, but again, you don't transfer a whole or half of an encyclopedia, but you transfer perhaps a part of a page, just one small chapter, and paste that, and then you have to find out whether that is satisfying the request of harmony, functional harmony. If it doesn't work, you just give up. You never publish that. But if you are lucky, the harmony is there, and you can further study it. These possibilities, I'm sorry. I wanted just to say here that we see here that in principle, what genetic engineering does is mimicking what happens in nature. Actually, the genetic engineers learned from nature how to do it. And therefore, the conjectural risk must be in the similar order of magnitude. And we do know that conjectural risks in natural biological evolution are quite small, and so are they in classical breeding techniques. And they must be of the same order of magnitude. I would like to say what is important for us scientists is to combine our reflection with ethical aspects. Do we want to do that? For example, I have less problem to bring a human insulin gene into a bacterial cell to produce human insulins than to really affect the human genome directly in the human being. So this is important. It remains important. Biotechnology, and that's what I mentioned already with the example of insulin. In the classical way, you had to use the organism as it is found in nature. You may try to improve by breeding. That has been done for decades, and random mutagenesis. Today, that's old-fashioned. And actually, it would also require, if you make heavy mutagenesis, in fact, all the other mutants which are not related to the selected phenotypes should also be studied, what they are. That has never been done, couldn't have been done before. In modern, explore the biological function, its mechanisms. If you like, you can try to improve by site-directed mutagenesis within the reading frame, or getting higher yields by expression control signals which are more efficient. And you can introduce, and that's the most important, the specific genetic information in the most appropriate organisms. You have just to be aware that it doesn't function all the time. This is what I had shown 4 years ago at the International Conference on Biotechnology in China in Dalian. I just mentioned that. I think that at longer term, one could try to, and that brings me back to agronomy, which was the field of Jacques. I think here is something important to learn. Getting food plants for feeding mankind, which are themselves of good health during growth and storage. Then, secondly, of high nutritional value for us who eat the plants, providing a healthy, well-balanced nutritional content for daily diets. My example is the golden rice, but you can imagine with our knowledge, increasing knowledge on metabolomics and all the genetics and proteomics, you eventually will have a certain number of things which you can use to ameliorate 1 or 2 dozens of natural food plants. At that moment, you have to reflect, where do you give priorities? That's a priority for me. Animal feed has less priorities for me. Nowadays, many of our agricultural crops are eaten by cattle, and we eat finally the meat, but it's a loss of energy, and you have to be careful. We are limited in water, fertile soils. We want to preserve biodiversity, and we don't want to affect too much the climate. So for sustainability, that is actually helpful very much if one does it carefully and with high responsibility of what one does. I give low priorities also to biofuels, crops for commodities like cotton, bioplastic. The problem is the human population, which we grow still in population density, and we have to reach an equilibrium with nature. Otherwise, we destroy our planet and future. The problem is, we think in terms of 100 years. Evolution thinks in terms of million years. And the sun is still providing energy to the planet Earth for long periods of time. So if we think on sustainable development, we have to think in long-term effects and not abuse too much what is presently still available, but we will run out soon if we continue as we are in the process to do so. This is a slide which I had devised. This is kind of summary and comes back to the introduction of Jacques. Our scientific knowledge, in fact, is obtained by efficient research strategies, originally observing, descriptive, more and more experimental, often disturbing the system to find out how the thing works, genetics, for example. And these have two kinds of cultural values. First of all, like knowing that nature cares for biological evolution, that I carry evolution genes and so on, for me is a worldview enrichment, and it provides to my orientational knowledge when I have to come to some decisions, a basis, and this is quite important. And one of the requests which the civil society may and would like to have is sustainability of that development, as I just mentioned. If you then, the second cultural value is the possibility to through application-oriented research, to come to practical technological use, which is often invasive and finally contributes to a shaping of the future. If you take care of sustainability, I think it's important to call on the really good power of technology assessment. And if you take decisions on the political level by civil society, you could also have policy assessment in the same way on the basis of our actual knowledge. And we should take more care of be very careful on shaping the future, not only for our children, but for long periods of time of life and biodiversity and diversity of habitats on our planet. Thank you for your attention.
Thank you very much, Werner. It was a wonderful lecture about the development of modern biology, genetics, and finally also some elements which are important for our life on Earth and other elements which might be important for keep us healthy. Thanks a lot.